everybody. I am here with my husband, Mr. Freeman, and we are going to show you the procedure for how to extract DNA from either the banana or the strawberry. And I thought about after I watched the videos again that some of you might think, well, I don't have all the things that they have in the video in my kitchen. And so I wanted to show you I don't have all those things in my kitchen either, but we can make it work still. There's no way you can do this wrong. So hopefully you might have a banana at home or maybe strawberries. If you have fresh strawberries, they're going to be a little harder to mash. I have frozen strawberries for daiquiris, but I pulled two out and I've thawed them a little bit to make them soft and mushy. And then if you have a banana, bananas are already soft and mushy. And so the first thing it showed you to do was take your banana and put it in the Ziploc bag, or I have my strawberries, they were fine here in the Ziploc bag. And then just start to mash up that fruit and just mash it as good as you can. Bananas, if they're super ripe, are pretty easy to mash here. And if they're not, you can just work a little harder, but you can just peel the banana, put it in there, and then make sure that you seal it back up. And so then you just go to work really kind of mashing up this fruit. So every living thing has DNA in its cells. And what we're trying to do is make sure that all the cells of the strawberries and all the cells of the banana are now kind of separated from each other. But what we haven't yet done is gotten the DNA out of those cells. So the better you mash it, the more DNA you're going to be able to get to come out of all those separated cells. And so sometimes you can just take the bottom of a glass and really mash good, especially the strawberries. And if you have fresh strawberries, you're going to need to maybe use something like this to really help grind up those strawberries and get them as mushed as possible. Now, how's your banana doing?
It's good.
Okay.
Mushy.
All right. So now, once we have the banana or the strawberry all mashed up, really ushy-gushy mashed up, then what we need to do is get the DNA to come out of all those separated cells. And so we're going to add to our Ziploc bags a solution that will cause the membranes to break open. And I hope maybe you remember that the cell membranes are made mostly of lipids, right? And so at home we use dish soap to get all of the grease off of our pans and off of our plates. This is the same stuff that's going to break open the lipids of the membranes around all those cells. And so we're going to add into our bag each 2 teaspoons of the dish soap, right? So here's, and again, there's no real exact, if it doesn't all come off of there, just, that's okay, right? So there's 2 teaspoons, and then maybe I can get you to hold mine. 2 teaspoons of into our strawberry. And you know what? The truth is, if you don't have strawberries or bananas, you can give this a try with any fruit you pick, right? If you've got any fruit at all sitting in your freezer, because we can't get to the grocery stores readily now, and all of that, any fruit is worth, I'd like to see what you get. And then we're going to add some warm water to both bags. There's your warm water. Half a cup, it said. So I'm using our warm water here. Thank you. And then we need to add a half a teaspoon of salt to both bags. So, this one, is this one? Ooh, my salt's a little chunky. There we go. There's half a teaspoon. There we go. Half a teaspoon. And now here's the next step that's just got to be a little patient with this. Make sure you seal that Ziploc bag back up. And then we're going to mix all of this around. We want all of that liquid and salt and soap to really work around all of these cells. And you're just going to try and gently mix it. We don't want this to get too bubbly. The soap solution will generate some bubbles. Some is fine, and even if you get more bubbles, oh well, it'll still work. but we're just trying to very gently mix all of that salt and soap and water around all of the cells that we mashed up with our strawberry or we mashed up with our banana. The reason that we have the salt in there is, and this one I don't think we got a chance to talk about before we had to all go home, is that all of the DNA is wrapped up around proteins inside these cells. And the salt will cause those proteins to unfold. If you remember the enzyme lab, when we heated that enzyme, it unfolded. And that caused it to not be able to do its job. Well, salts could do the same thing with proteins. It'll cause them to unfold and let go of the DNA. And now we've broken up the cell membranes and we've unfolded the proteins that were holding them in place. And now the DNA is going to come out of all of those cells and into this watery solution. And so you just gently work that around. And if you're at home, I'm talking to you, so that's passing the time, but if you're at home, you can just kind of think about maybe 3 minutes or so. Now, once you have that, your DNA is floating around in the liquid of the solution. The cell walls and the other portions of the structure of the banana or the structure of the strawberries, is all of the chunky stuff that you see in there. We need to separate the liquid from the cellular material. And for that, they have you using a coffee filter. Now, coffee filters, a student asked me if we could use paper towel. I'd say go for it. Just be really gentle with the paper towel. But I don't have a funnel at home, just like you probably don't. So what I did was I cut a water bottle in half, and we cut a pomegranate juice bottle in half to create some place where we could set the coffee filter. And then once you have that in place, again, I don't have beakers at home either, so I have a glass cup and I have a flower vase that I'm using to collect the liquid as it drains through our coffee filter. And so this one's a little narrower, so you have to pour a little careful. What you're gonna do now is pour all of this liquid out of the bag and into this coffee filter and let the liquid come through the coffee filter. All the cellular material will stay on top, and when the liquid that comes through the coffee filter is filtered, it still has the DNA. The DNA is in solution in that liquid. So we have to be patient with that a little bit, pour a little slowly, and just let the liquid drain through your filter. If you use paper towel, same thing, just be really gentle with the paper and it probably will work. And so you see, oops, yep, it's coming through there. So let's see, we might very gently sort of move this around and see if we can get some of the banana mush to get out of the way so the liquid can come through. Do we have any liquid draining into the bottom? Very slowly. Okay, let's pour a little more